3D printing techniques have been used by doctors to give personalized treatments for patients throughout the years. With technological advances, bioprinting stem cells for tissue engineering has the potential to play a pivotal role in alternative treatments. The demand of transplantations for patients with end-stage organ diseases have increased over the years and keep climbing. The biggest problem for organ transplantation is organ rejection. This is where the immune system recognizes the new organ as foreign and begins to target the cell, acting as if it is an infection. To prevent organ rejection, patients need to take drugs to suppress their immune system. This makes them more prone to infectious diseases like cancer. And even with these drugs, the average lifespan of a transplanted organ is only about 12 years, and the patient may require multiple transplants throughout their lifetime. Novel therapies are being discovered to print new organs from the patient's own cells that could potentially last a lifetime. Previous researchers have developed organoids in the lab by suspending embryonic stem cells and growth factors using 3D cell culture systems. A common challenge faced when using organoids is that the size is limited to 1 millimeter because nutrients can only diffuse across a small distance. In developed organs, blood vessels are needed to supply nutrients to the tissues, and these designs are hard to develop. A potential way of making these new organs would be through lab-grown, fully functional human organs. Bioprinting is similar to 3D printing, but uses a printable material that contains living cells that survive the printing process. This material is called bioink, which is composed of a mixture of living cells, hydrogels, target cells, and chemicals that ensure cell interaction. Specific biomaterials are placed in order to ensure the bioink reacts to particular activation methods. Bioinks have single or multiple cell types and hydrogels and chemicals for more complex structures. There are three main bioprinting methods that are commonly used. These can be referred to as extrusion-based, droplet-based, and energy-based bioprinting. Each have their own benefits of using depending on the type of treatment you are developing. Extrusion-based bioprinting is most similar to traditional 3D printing, where bioink is loaded into a nozzle and pushed through a tip. It can be very precise and print at 400 microns in diameter. These structures can be stabilized onto flat surfaces or into liquid medium that promotes stabilization. This method uses dynamic pressure and mechanical pressure through pistons or screws to force droplets of bioink out. This process can kill cells if the nozzle is too small or if the printing pressure is too high. Depending on the bioink material used, the printed structure can become stiffened or it may require additional stimulus through UV light, chemicals, or undergo physical processes. The second type of bioprinting can be considered as droplet-based bioprinting. This method deposits droplets of bioink on the desired surface. This category can be broken down into three subcategories of inkjet-based, microvalve-based, and lift bioprinting. Inkjet is similar to normal printers as they work in either thermal or piezoelectric systems. Thermal mechanisms heat up a small surface of bioink. Piezoelectric currents are sent through the bubble to make pressure and drop bioink. Microwave-based bioprinting involves small valves that can be controlled by electromagnetics to release bioink that is under air pressure. Laser-induced forward transfer, or lift bioprinting, uses lasers to accurately position cells on a substrate. This method uses lasers, lens, ribbon, and a substrate. The ribbon has a gold coating that is activated by the laser to print bioink on that substrate. Lift bioprinting is very precise, but it is expensive and time-consuming. Lastly, energy-based bioprinting uses a focused energy source to stabilize biomaterials and solidify the bioink. Stereolithography is when a laser is used to harden small amounts of bioink that contains light-sensitive hydrogels. This method is different from other forms because the biomaterial is already in place and the laser is used to stabilize the bioink. This method is currently being used by Dr. Chen from UC San Diego. He implements this bioprinting technique by using light to activate bioink to make intricate designs like the heart. Scientists are 3D printing fully functioning organs by creating tissues and organoids complete with blood vessels. One of the main challenges is how to supply oxygen and nutrients to all the cells within a fully sized organ. By using focused light to activate bioink, Dr. Chen's research has been able to develop functioning bioprinted cells. When printed correctly, the stem cells in the synthetic tissue behave similar to normal tissue by interacting, exchanging nutrients, and multiplying. Some treatments are in clinical trials that can print structures within 30 minutes. For example, scientists are developing handheld 3D bioprinting devices that are being used in orthopedics to print meniscus, optometry to treat ulcers in the eye, and trauma therapy to print skin for burn victims and trauma patients. The best successes are with structures that are flat or hollow. Bioprinted human bladders are commonly used. Researchers are developing ways to incorporate blood vessels with bioprinted tissue and rat models to promote facial nerve regeneration. Scientists designed a 3D printed silicone heart, but it was unable to survive long-term due to mechanical failure. 3D bioprinting techniques with stem cells have been used in patients for a variety of treatments. Previous methods to extract bone marrow stem cells from patients included performing invasive techniques. With advancements in stem cell technology, scientists are able to engineer adult skin cells and create induced pluripotent stem cells. This overcomes ethical issues of using embryonic stem cells and can design more personalized therapies with results more close to the human biology. Bioprinting was recently used to combat COVID-19 by mass-producing human brain organoids to test against the virus. 3D printing with stem cells can play a large role in drug testing instead of using animal models. Human tissue, skin, and cartilage have been bioprinted, as well as organoids of the lung, kidneys, and hearts. These all are great resources in surgeries and for drug development. They are currently bioprinting stem cells and tissue engineering for ears, livers, bones, and designing hearts with patients with their own cells. Israeli scientists at Tel Aviv made a 3D printed heart with the patient's own stem cells that function properly. At Rice University, they bioprinted a heart that is designed with a vascular network that is similar, that uses natural pathways of blood, oxygen, and other vital fluids at the same small scale. Vascularizing has always been a challenge for 3D bioprinting small organs, but researchers use stereolithography with organic food dyes to shoot light at areas of the bioink to solidify tiny designs. With the ability to 3D bioprint stem cells into complex organoid systems, I believe the most useful implementation is testing for safety and efficacy in drug development. Drug testing with animal models is outdated and is not required when using 3D bioprinting stem cells. Bioprinting provides a more accurate method of testing when developing new drugs and treatments because they conduct tests on bioprinted human organoids instead of murine or non-human primate models. Bioprinting can help save lives for those who require organ transplants. I believe off-the-shelf, pre-made allogenic bioprinted organs is the future, but this can only become a reality if researchers continue to push technologies and discover novel techniques. By using hybrid bioprinting methods in stem cells, organ development can become specialized and efficient to help reduce the amount of patients waiting for a potential owner. With new stem cell technology, 3D bioprinting organs can be grown from the patient's own cells, and eventually we will have the ability to bioprint personalized organ treatments for individuals. 3D bioprinting stem cells have proven to be great in the field of tissue engineering for making biostructures like skin, bones, organs, and orthopedic tissues. To face challenges like vascularizing organs, new techniques are invented to mimic similar characteristics of biological, structural, and mechanical properties in bioprinted organs and tissues.